Children, I hope you have gone through my previous videos and done all the work that was assigned to you. Let us now proceed further. Children, today we are going to start the chapter Chemistry in Everyday Life. Chemical compounds are certainly attached to our lives. Our bodies get energy from the compounds obtained from the food which we eat in the form of sugars. Our clothes are made up of organic compounds. We take drugs when we are ill. In modern time, chemistry is being applied to produce materials for space travels, fuels, and rocket propellants. All sorts of building materials, synthetic fibers, microchips for computers, explosives, and chemical weapons. Now, first the most widely used chemicals is in the production of drugs. So let us see what are drugs. Drugs are the chemicals of low molecular masses between 100 to 500 microns whose intake by a living system has a physiological effect. If the drug helps in treatment of disease, then we call it as medicine. The branch of science dealing with the treatment of various diseases with suitable chemical substances is known as chemotherapy. Overdose of any drug can be toxic and poisonous. The drugs, they are classified into 4 types. First is on the basis of pharmacological effect. This is based on the effect of drug on biological systems. For example, antibiotics restrict the growth of microorganisms and prevent the spread of infection. Second is on the basis of drug action. This is based on the effect of drug on particular biochemical process. Third is on the basis of chemical structure. Drugs with similar chemical structures have similar action on biological systems. For example, sulfonamides. Fourth is on the basis of molecular targets. It is based on interaction with biomolecules such as carbohydrates, lipids, proteins, and nucleic acids which are present in our body. Now let us study first about the drug target interaction. Now you know that the drugs, when administered to a living system, interact with the biomolecules. Proteins which function as biological catalysts, they are called as enzymes. Those which are involved in receiving communication signals, they are called as receptors, and those which transport polar molecules across cell membranes are called carriers. When a drug is administered, it may target any one of the biomolecules, and the therapeutic effect results from the interaction between the drug and the biomolecule. Now, we'll study now about enzymes as drug targets. Now, the first is catalytic action. In the catalytic activity, enzymes perform 2 major functions. First is the enzyme binds the substrate, which is the reactant or the drug, and holds the substrate molecule at active site. The enzyme and the substrate combine to form enzyme substrate complex. Now see here, suppose if this is the enzyme and this is the substrate. So the substrate gets fixed into the enzyme, this place over here, and it forms a enzyme substrate complex. This bonding may be by van der Waals forces, electrostatic forces, hydrogen bonding, etc. The second function of an enzyme is to provide functional group that will attack the substrate and carry out chemical reactions. So this is how the drug acts. See, for the enzyme will capture the substrate and then it will provide the functional group which will attack the substrate and the chemical reaction will take place. Next is drug enzyme interaction. Drugs inhibit the attachment of substrate on active sites of enzyme in two different ways. The first is drugs compete with natural substrate for their attachment on the active site of enzymes. Such drugs are called as competitive inhibitors. Like see over here. This is the enzyme. This is the drug which we are providing from outside, and this is the natural substrate which should actually fit into this enzyme. But before the substrate can fit into the enzyme, the drug comes and fits into this place, and so it inhibits or prevents this substrate from getting attached to the enzyme. So such type of drugs, they are called as competitive inhibitors. The second is some of the drugs, they do not bind to the active site of enzymes. They bind to different site of enzyme which is known as allosteric site. This binding of inhibitor changes the shape of active site in such a way that substrate cannot recognize it. See here, this is the enzyme. This was its original shape. Now when the drug comes and combines over here at the allosteric site, this is the allosteric site. So this drug binds over here, and what it does, the shape of the enzyme which was earlier this now becomes this. So now even when the substrate will come, since the shape is not appropriate, it will not be able to attach to the enzyme. So in this way, the drugs will inhibit. Now when the drug enzyme interaction involves the formation of strong covalent bonds, the binding is irreversible and the enzyme is permanently blocked. This enzyme inhibitor complex then gets degraded and a new enzyme is synthesized. So this is how the drug enzyme interaction takes place. Drugs inhibit the enzymes from performing their activity. Next is receptors as drug targets. Just a minute. Receptors as drug targets. Now the receptors which are present in our body, the receptor, they are proteins. They are receptor proteins. They are embedded in the cell membrane in such a way that the small part having active site projects out of the surface of the membrane and opens on the outside region of the cell membrane. See, this is an animal cell diagram over here, and from here we have cut a section of the cell membrane and enlarged it. So on enlarging it, you can see over here, this is the cell membrane, and here this is the receptor protein, and this shape over here, this is the binding site of the receptor. Okay, so this is projecting outwards, and this is the interior of the cell. So small part of this active site is projecting out of the surface of the membrane, and it opens on the outside region of the cell membrane. Now in the body, message between two neurons and neuron and muscles is communicated through chemical messengers which are received at the binding site of receptor proteins. To accommodate a messenger, shape of receptor site changes and brings about the transfer of message into the cell without entering into it. The receptors show selectivity of the chemical messengers and act by receiving them at their active site. See here, this is the chemical messenger which is coming near the binding site. Alright, so this is the outer surface of the membrane, and this is the binding site, and this is the chemical messenger. So the shape of the receptor, it changes to accommodate the chemical messenger. Okay, and after this, after the messenger has got attached to it, the shape which was like this earlier, it has now become flat at the bottom. See, so the shape of receptor site changes, and this brings about transfer of message into the cell without entering into it. Now again, the receptor is showing selectivity of chemical messenger. Not every messenger will be received, and they receive them at their active site, following which their shape changes, leading to transmission of message without physical relocation of the receptor protein. So this is how receptors act as drug targets. Next, you have to study about antagonist and agonist. Drugs which bind to the receptor site and inhibits its natural function. They are called as antagonist. They are useful when blocking of message is required. Drugs which mimic the natural messenger by switching on the receptor are called as agonist. They are useful when there is a lack of natural chemical messenger. So this is all for